So today we're going to talk about molecular genetics. We've already been talking about molecular genetics just by simply talking about the structure of DNA. Now the structure of DNA, as you remember, there's a phosphate deoxyribose backbone, and then there's the nitrogenous bases that make up the latter. And that is also the coding section. And we keep using these terms, coding section and things like that. But we haven't really talked about how this works. So today I'm going to introduce the entire process from DNA to a trait. Now I understand that you may go, wait a minute here, this is really fast. We're going to break it down afterwards, but I think it's good to see the entire picture, and then we'll go back and look at sections of it. So for our first bit of homework, we're just going to learn about the whole story. So how are different types of cells created and maintained? This is a good place to start because we keep talking about DNA as a blueprint, but we need to figure out, now wait a minute, if there's only one blueprint, why does this red blood cell look nothing like the cell right next to it? Kind of a crazy question. So let's look at this. What is making this happen? Well, the answer to that question is differential gene expression. Differential gets to that one gene is expressed in one kind of, not just one, but certain genes are expressed in one kind of cell, and in another kind of cell you're going to have other genes expressed. Express means to show itself. So not all the genes are expressed in every cell. And that is really key to having one code, yet every cell having different abilities to do different things. So let's think about this for a bit. In all 50 trillion of your cells, you have the same genetic information, whether it be a red blood cell or a skin cell or a brain cell, those genes exist. However, different cells use the same blueprint in different ways. So that same blueprint has the whole suite of genes, but only certain genes come out depending on which of the cells you are. And how does this work? Well, in essence, the control of gene expression occurs by regulating the flow of information of DNA to protein. So this next section we're going to be talking about, DNA going to protein, is going to be how we get some cells that are going to be one type of cell and another, and skin cells being skin cells, and liver cells being liver cells. That's how this works. So you should get this down. This is called the central dogma, and that means the central idea or the main idea of molecular genetics. Molecular genetics has this main idea, and in the picture here you can actually see this. And this idea is this, that you start with DNA. You can see there in the nucleus in the color red, it's labeled DNA, and DNA will then go to RNA. Now there's a couple steps in this, and we will be talking about these steps, but you have mRNA, which stands for messenger RNA, and then that leaves the nucleus. You can see it leaving the nucleus outside of one of the nuclear pores into the cytoplasm, and it goes to a rRNA or a ribosome, and then it meets up with tRNA, and then from there you get protein. So this main idea, DNA to RNA, RNA to protein. If you don't have a good way of learning that, say it out loud 15 times, because this is the mantra that we want to make sure you know. And that protein is what gives you a trait. Okay, so that's what we're talking about here. Going from DNA to RNA is what we call transcription. And that's because DNA it is a language of A, T, C, and G. And that is very similar to the language of RNA, which is A, U, C, and G as the components of the code. There's only just one difference in the nitrogenous bases, and there's one little difference in the sugar. But that's the only difference between DNA and RNA. So if I transcribe something, for instance, I will say, write what I am saying out loud. Well, I'm speaking in English and you're writing in English. You are transcribing what I am saying, or doing transcription. But the second part is called translation. And translation is where you take one language, the nucleic acid language, and change it into a protein language. And we'll be talking about how that works. RNA language or nucleic acid language to protein language. So, transcription is a key step in gene expression. Transcription is where part of the DNA is found, and that part is the part that is going to be made into a protein. So that is one of the key steps in understanding why we can have so many different types of cells and only one code, because where that mRNA is made and where that transcription takes place determines what kind of trait we're going to have. So transcription makes an RNA copy of DNA. So RNA. There is, as I said, two differences with RNA to DNA. RNA is a nucleic acid polymer that uses a slightly different sugar than DNA and the base uracil in place of thymine. So make sure that you have this down. Look at DNA, look at RNA. You can see in the RNA, which is in the yellow, on the carbon there's an OH, or what we call a hydroxide group. And in the DNA, deoxyribose, there's not an oxygen on that same corner of the deoxyribose. So that's the big difference with the sugars. But then you have the base uracil versus thymine, which is the other difference. So there are two differences between RNA and DNA. RNA is largely single-stranded. So there are exceptions, but largely when we think of RNA, it has one strand. DNA, remember, has two strands. There are different RNAs with distinct functions. So the first one is messenger RNA. It's found in the nucleus, and it migrates to ribosomes in the cytoplasm, and its function is to carry DNA sequences information to the ribosomes. The next one is transfer RNA, or tRNA. It's found only in the cytoplasm. It provides linkage between mRNA and amino acids and transfers amino acids to ribosomes. And then you have ribosomal RNA, or rRNA, and it is also only found in the cytoplasm, and its function is structural component of ribosomes. So it actually makes up what a ribosome is. And so these are our three key types of RNAs. There are other types, but we will not be covering them in this class. So the other ones are microRNA, and there actually has been other research that has been done as well on RNA, because it comes in a number of forms. So transcription. What happens is you have the DNA opens up, okay, and remember we saw DNA opening up when we talked about replication, where we make a new strand of DNA. This is where now we're going to actually use DNA as a blueprint. In transcription, you can see the C's pair with G's, just as they did those. You have the RNA polymerase come in and bring in these nucleotides to make a strand, and it uses the blueprint of DNA to make the RNA strand. The enzyme RNA polymerase opens the DNA strand and synthesizes an RNA complementary to only one of the DNA strands. Then, in transcription, that RNA strand gets longer and longer until it comes to its end point. And the decision to transcribe a gene is the most important step in the control of gene expression. So once it's determined, hey, we're going to be using this gene, that is the key to genes actually being the one that is expressed in a particular cell. Transcription starts and stops at distinct sites at the end of a gene. So that is that length there of the DNA, that within the entire long piece of DNA, that is a gene. So, as you can see, the genetic language uses 4 letters written into 3-letter words. Let's look how this works. So the DNA in transcription makes mRNA, and mRNA makes a long chain. But then to get that into protein, you take that and you chunk it into 3-letter sections, and those 3-letter sections relate to a certain amino acid that is then added to a protein chain. So going from the DNA to the RNA, that's transcription. Going from the RNA to the protein, that's translation. So what with the amino acids, what is this code we're looking at here? Well, an amino acid, and we talked about this a little bit at first semester, you have an amino group, that's the NH3 on that side, and then you have the carbon side where you have the C with a carbon double bonded to an oxygen and another bond to an oxygen. Then the carbon, remember, carbon can take 4 things it can attach to, and that's one reason it is the structure of all living things. Then we have a hydrogen attached to it, and then we have an arm. And that arm, which is highlighted on both of these examples, glycine and isoleucine, is what makes an amino acid unique. And there are 20 different unique amino acids, meaning that arm is unique with the 20 different amino acids. So here we have glycine, it just has another hydrogen. And then you can see that long chain in isoleucine. And then you have those 20 amino acids, and that code is the code that a protein is made from. So what translation accomplishes is having a great big long chain of amino acids, and we call that long chain of amino acids a polypeptide chain. Lots of chains and trains here in biology. So in translation, information present in the mRNA is read by the ribosome to synthesize a polypeptide. And we'll be talking about the details later, but that's the main part of the story. So the sequence of amino acids determines the structure and therefore the function of the protein. And translation is complicated, and we'll be talking a little bit about it. And it requires ribosomes, mRNA, tRNA, and amino acids. So when we talk about what antibiotics do, the way they are able to kill bacteria is they can block steps in translation within bacterial cells, and that can kill those bacterial cells in your body, and that can be helpful to you if you have a bacterial infection. We have tRNA, and it's an adapter that couples the codons and the amino acids. So what tRNA has is it has what's called an anticodon on it. And that anticodon, that little part at the bottom, you see it labeled there. tRNAs are my favorite because they have that cute little shape. It then attaches to the mRNA at the matching point. And that anticodon and that codon only reads for one specific amino acid. So it always brings in the correct amino acid that way. Here's the genetic code, and it's basically biology's Rosetta Stone. Knowing which codon sequence matches up with which amino acid is the key to being able to read the genetic code, and we'll be talking about that a little bit later. So ribosomes are complicated proteins that are synthesizing machines, and they are the location where all this takes place. And then translation is a cyclic multi-step process where amino acid after amino acid is brought into the process, one after another. And so you can see one amino acid comes in with one tRNA, and then the next one comes in, and then the next one. So the basic genetic mechanisms are universal: the storage of genetic information in DNA, the use of an RNA intermediate that is read in 3-letter words, and the mechanism of protein synthesis are essentially the same in all organisms. This is key. This central dogma of biology, this is not just for animals, this is not just for plants, this is not just for fungi, this is not just for protozoans. This is also for bacteria, this is also for archaebacteria. All organisms have this same universal genetic mechanism. And among other things, this means cancer can be studied productively in flies or yeast. When you wonder, why do they do animal trials? Well, that's how they can do animal trials, because if it's the same DNA and the same basic mechanism that's happening in you and happening in that fly, then these first initial steps of medical research can be done on animals before we just try them out on humans. Now, there's some ethics around all of that, but people are able to use these systems, and it definitely is done. It also means that human genes can be expressed in plant, mouse, or mouse genes in a yeast. So what we can do is we can actually take our genes, put them into another organism, and that organism can express that protein that was expressed in a human. So here is a tobacco plant expressing the firefly gene for how it glows in the dark. So this is our basic start to molecular genetics, and we'll be taking some time with those details. Make sure you do get the notes for this, however.